Do we need to wait a little bit longer or should I get started?
I can briefly check against the attendance list to see, but
Okay,
it does
sounds good.
seem, yeah.
Yeah. Thanks a lot, Amber.
I do think we have a few more registered than are here now, but I think you should be fine to get started at this point, or since we're 5 minutes past.
Okay, yeah, I can go ahead and do that. So, hi everyone. My name is Amber. I am one of the research computing facilitators here at the Open Science Pool. Danny is my colleague, and he'll be running the majority of the training portion. But we're all really happy to be here to show you what you can do with the OS pool and answer any questions that you have. Um, and so without further ado, I'm going to go ahead and get started. So today's presentation is about scaling out your research, running your workloads on the Open Science Pool, and using the Open Science Data Federation. And so really what drives the Open Science Pool, which is this national resource that you can have access to as a researcher, is this idea of high throughput computing. And we like to describe high throughput computing as a way to get a lot of work done by using a lot of resources. We want to solve researchers' problems. So, for example, you could be a researcher who has a problem where you need to extract some sort of feature from a bunch of different images. And that is a task that you can do on your laptop. But when you have a lot of images, that becomes a bit of a larger task, right? Or maybe you're doing some sort of machine learning workflow and eventually you want to run inference on certain documents. Perhaps you have a simulation with lots of parameters that you want to try, or you have genes and you want to do some sort of comparison. So one thing that all of these problems have in common is that they involve a list of tasks, and that's where high throughput computing really comes in. So our founder and director, Maron Lively, says that the ideal high throughput computing workload is when you have a list of jobs. And so the PATH project, which is kind of this project that the Open Science Pool lives under, provides services that can advance the science of researchers at all campuses who have computational workloads that consist of a list of jobs. And so what that looks like is that when you have that list of tasks, you could run it on your computer, slowly run it in a serial fashion, but a way to speed this up is to parallelize these tasks by running them on multiple computers. So in essence, it's like we're giving you keys to a computer lab, and then you're able to run your workload on every single computer in the computer lab. So that is kind of the idea of high throughput computing, except it's not quite so physical where you're going in and into a computer lab, signing in. Instead, we do this all over remotely. So how it works is that you will have an account on what we call an access point. You might hear a login node or a submit node. And this is where you're able to access the computer remotely and then upload your data and your software here. Any US-based researcher can get access to an access point when you apply for an Open Science Pool account. You'll create a job submit file. So this is a text file that describes what kind of workload you want to do. Say you want to run multiple inferences or do multiple gene comparisons. And then you submit your job to our system, the Open Science Pool, through a software we call HT Condor. So HT Condor is this job scheduler that kind of manages researcher jobs all across the United States. So HT Condor is going to get information about the workload you want to submit. It will find a computer out in the Open Science Pool. And so these computers are could be at any institution across the United States, as long as they decide to donate their resources to us. And then your jobs will land on those machines and run there. So once your jobs have run, any outputs they create are then transferred back to you at the access point. And so in this kind of schematic, you can imagine that it's very automated. It's sort of non-interactive, but in this fashion, you can kind of see how you can scale up your workflow by not just creating one submit file for a job, but creating a submit file for many jobs that can run on many computers. So this is sort of a general way of idea of how the Open Science Pool works. And with that, I'm going to go ahead and turn it over to Danny, who will show you how the system works with an actual hands-on tutorial.
Thank you. Great. So I will share my screen. All right. So for today, we have two tutorials, sort of hands-on tutorials planned for you guys today. First one's going to be a sort of canonical high throughput bioinformatics example called read mapping. And then the second tutorial is going to be using the OSDF, the Open Science Data Federation, in order to manage and share your data with yourself throughout many of these machines throughout the US, but also with other colleagues that you might want to share your data with. And so with this example, we usually like to get started with sort of getting to understand what the researcher or who the researcher is. And so this is a very typical biology researcher scenario where you're a new research student at a lab. Let's say you're a PhD student that's working in a marine bio lab and you're studying humpback whales. And you are studying their genomic evolution. You're trying to understand how genes are evolving. And so you've been working on setting up a pipeline to take genomic reads, the little pieces of DNA that have come out of the sequencer, and compare them to a reference genome that's available online. And so you've been able to do it fairly well on your own laptop, maybe a lab workstation, for a few hundred, maybe a couple thousand reads. But now you just got a massive grant. You've sequenced the genomes of tons of whales. And so you want to compare across a larger pool. And now you have a problem of having too many reads to actually map on your own workstation, your own work laptop. But luckily, our researcher knows that they can scale up their research using the OSPool by breaking down that problem into a list of jobs. And that's really the crux of what we're going to be getting at for this tutorial. And so diving a little bit deeper into this sort of problem, when we are mapping reads, for folks that don't have a background in bio, you're usually having sort of two main data files. You have a reference genome that's usually very well curated over many years, that you are taking those little pieces of DNA and essentially trying to do a matching of each piece to the reference genome. Now, these reads come in these files called FASTQ, And so you'll have millions of reads in these files that can very quickly just become a problem of too much data. And so for today's example, we're going to break that problem down in 3 steps. We're going to first start off by splitting up our millions of reads into smaller, about 1,000 set subsets. We're then going to submit a single simple htcontrol test job just to make sure that we understand how things are working. We can submit jobs. It's a good sort of wellness check before you start scaling up. And then we'll actually start scaling up our research so that we can submit all of our reads throughout using the OSPool. And so when I say that mapping reads is one of those canonical HTC problems, the core of it is with this FASTQ file, this set of reads, typically you might be looking at something like 5 million reads in a single file. That can easily start to look at 100 gigs, 200 gigs worth of data. And so if you wanted to map this all on one machine, you'd have to find a machine that has enough memory, so enough RAM to read all of that in, but also enough disk. You have to have the right piping in terms of network so that you can move 100, 200 gigs of data back and forth. And so it very quickly can scale even beyond what a nice HPC can handle internally. But luckily, reads are one of those things that it doesn't really matter if read 1 maps in one place or independently from read 2. We just want to see where they're mapping to on the genome. So we can split those apart and send them out through hundreds of machines and run them in parallel. And so that's sort of the name of the game with the OSPool. We have these massive problems, like we've been saying. For this case, it's too many reads, giant file. And if we can split that and do sort of a divide and conquer workflow, we can take that giant FASTQ file, subset it into, in this case, let's say 20 FASTQs, and submit each one to a different machine across the US. And so they all run semi-independently and parallelized, and you'll be able to process your data as a whole much faster than if you tried to process it in series on a single machine. It also allows you to hit a lot more machines. So imagine trying to find that 200 gig machine. That's going to be a rare find in a network. However, you can probably find a machine that has at least 1 gig of disk and 1 gig of memory. Most of our phones have more than that. And so because we have this distributed network, the smaller your resource requirements, the more constrained your inputs are, the more machines you're going to be able to match to, and the faster your jobs are going to start. And so we can now that we understand a little bit about the problem, can actually get our hands dirty and get started on it. So we have this link. If you go to osg-htc.org/notebooks, you'll be taken to a web page that looks something like this. As welcome to the OSPool notebooks. There's a little orange button that says sign in with CI logon. It'll take you to this green looking page. You'll be able to sign in with your institutional ID. So look up your university and it should show up in that list. And then once you sign in, you'll have 2 options. Today, we're going to be working with the guest account. So you don't have to have an OSPool account already made. This is sort of a guest account that you can use for the tutorial. And so once you're logged in, you'll see a page that looks something like this. And so from that, we'll actually get started with the tutorial. I'll give folks about a minute or so to get started. And if you have any questions, you can also just pop them on the chat or unmute. And so you'll see that it'll take a few minutes to load up. So my notebook's loaded. This notebook is going to be available also beyond the training. So if you want to go through it again afterwards and dive into some of the other tutorials that we have a fast QC tutorial, we have a Pelican tutorial. Those are really good resources to go through and play around with. It doesn't have access to the full OSPool account, so this is a smaller subset. But as you start to play with the notebook and you think about your research, if this is something that you want to continue pushing forward, we're always happy to chat more and get you an account on the OSPool. But for today's tutorial, right now we're going to focus on this Minimap 2 tutorial. So we double click it, you'll see this orange button. I think we should be able to sign in around 30 or 45 people. If you're getting an error message saying that you're not able to sign in, just drop it on the chat and we can message the sysadmins. We should see this orange icon on the left. That's going to be our tutorial Jupyter Notebook. And so we're going to go through this. Jupyter Notebook, if you aren't familiar, is a really nice markdown integrated way to integrate some of the command line arguments that command line commands that we're going to be putting into our actual terminal, but also surrounding it with markdown so that you can have a little bit more information, pictures, things like that. And so all of the commands that you see in these gray boxes, you can enter them directly on the terminal. If you'd rather work on the terminal, there's a terminal option down here. But for the most part, for this tutorial, we'll be working within the notebook service to keep things neat. So now that we're inside our notebook, we're going to want to make sure that we actually change our directory to this tutorial Minimap 2 directory. And so we can do that by clicking the cell right here. And there's a play button up at the top that you can click. And so if you click the next cell that says PWD, it should print something out like home/jovian/tutorial-minimap2. And so that tells you that, yeah, you're in the right directory. And so if we check out our directory, these two should be matching about the same. They're ordered slightly differently. But we see that, yep, we're in our tutorials Minimap 2 directory. We have our Minimap Jupyter Notebook. And we have a couple directories in here that are empty. So these are pre-made for you when you launch the notebook. And so in here, we're going to start adding our data. We're going to start copying our files over so we can actually start working with it. And so we have this download_data script. If you go ahead and hit play, it'll start downloading your data from our shared directory. This will take a few minutes, so I'll chat a little bit more about sort of our data structure that we're going to be looking at. So what our directory structure will look like as we move through the tutorial is something like this, where you have this tutorial-minimap2 directory that we're currently in. And under that, we'll have inputs, outputs, logs, and software. As we move through the tutorial, right now we're copying this humpback whales _reads.fastq file into our inputs folder. So if we check here, it's downloaded in there. And this is that really big file that we want to subset and divide. We also have a folder for our outputs. Eventually, once we submit this, we're going to submit about 70 jobs. You can imagine receiving 70 files coming back can be a little overwhelming to try and parse through if you're just dumping it all into the same directory. So we want to move them to subdirectories to keep things neat. You also see that we have a logs file or a logs folder. In here, we'll capture our standard out, standard error, so the messages that you'd normally see on your terminal if you were physically running these commands, as well as a htcondor job log. And so that'll capture what's happening with your job itself. One of the key things to remember is your jobs are going to be running all throughout the US. So you're not going to be physically located in front of the machine. So if something goes wrong with your job, we need to be able to capture that into a file that you can then parse through by logging into the access point, the login node, and reading that log file. You also see that we have a software folder down here that has this minimap2.def or definition file. We're not going to go too into detail today about how to set up containers and set up your software environment, but I can briefly go over this is how it works. We're going to be using an apptainer container. It's similar to Singularity if you're familiar with it. And in this case, we're taking a minimap or a miniconda 3 container that already exists on Docker Hub. And we're using a standard conda command to just install a few softwares, minimap2, samtools, and bedtools, which are the sort of workhorse programs of bioinformatics into that container. We also have a second directory that you're not going to really be able to see on the left-hand side. And that's this OSPool-guessap/data/Chauvin directory. And that's a special directory that is backed by the Open Science Data Federation, the OSDF. And so this is a really good place to place, or a really good directory to place large files that are often reused across a lot of jobs. It has some built-in caching. So as your jobs start to land across the US, you'll be able to, awesome. You'll be able to capture your file from a closer point. Let's say your job lands in South Florida. It'll try and download your file from, say, Atlanta instead of having to go all the way from Wisconsin-Madison, where you might be logged into. And so it just places your files closer to where you're going to be doing your compute. If you have any questions, also just feel free to mute, drop them in the chat. I'm trying to keep an eye out for them. So we can make sure that we got all of our data files. So we're going to ls our current software directory, and that's right here. We have that definition file that we were looking at earlier. And we can also ls or list our files in that OSDF directory. And in here, we see that we have this minimap2 SIF file, and that's that container file. So it's the actual image that we converted that DEF file that had the conda command into a final runnable binary file. And so what we're going to be focusing on today is how to create that list of jobs and run them using htcondor. And so, like I said, we have this problem right now of we have a massive FASTQ file. We want to subset and submit out through the entirety of the OSPool. And so the way that that would look like is, let's say you submit 100 jobs because you have 100 subsets. Job 1 will get subset 1, job 2 will get subset 2, so on and so forth. And it will process the same command essentially on each of your files, where the only thing that would be changing is that input file that you're giving it. And so it's a way to scale up and in mass process all of these hundreds of files at once. And so for Minimap, the command is relatively simple. It's a short command that we're going to be running. We're going to be running this minimap2, and that just says run this program with the AX function that just says map your reads. And it's going to be using a type of read called an ONT read. And so this is one of the newer generations types of reads coming from our newer sequencers. But what we really need to focus on is these three last parts of the command. So we're going to input a reference genome. And so that's going to be our reference genome from the humpback whale that's already been created and saved on your OSDF directory. You're also going to give it a path to your reads. And so one way that you could do this is just give it all of your reads right there as a single file. And that's going to take a really long time because you're going to have to parse all 5 million reads one by one serially. In our case, we're going to give it here our small subset of our reads. And then this is going to output what's called a SAM file. And this is a alignment file that Minimap2 gives us. And so in order to split our reads, if we go back to our inputs folder, we have this humpback whale underscore or humpback underscore whales underscore reads.fastq file. And so we want to split this up into about 1,000 reads per subset. It's just a good number to start with. And for those that aren't familiar, reads in FASTQ format are made up of 4 different lines per read. And so if we want to split them about 1,000 reads per file, you can use the split command in Linux, and it has the -L option, which just means line. And we're going to put 4,000. The reason that we're doing 4,000 is we want 1,000 reads per file, and each read is made up of 4,000 4 lines. So 4 times 1,000 gives you 4,000. We're going to give it this input file. So it's going to go to our input directory and read in our full FASTQ file. And then we're going to add a suffix at the end, which is just saying at the end of that subset file, just add underscore humpback underscore whale underscore reads.fastq. And at the beginning, we're going to append inputs so that it goes into the input directory /subset underscore. And so this might be a little abstract at the moment, but what that will eventually give you is these sort of styled of formatting, where you have subset underscore and then a unique identifier. In this case, it's going to be an alphabetical list that's going to increase throughout. You'll have subset AA underscore humpback underscore whale underscore reads.fastq, subset B or AB, subset AC, and so on and so forth. So if we run this command, it'll take a few seconds, but you should see a bunch of files on the left-hand side are generated. And so if you do an ls on this line, you'll see that you have your FASTQ up here. That's our original file that we downloaded. That's our massive file. And then we have these smaller subsets that we've now created. If you wanted to see how many subsets you created, you can use this command where you're listing all the files in the input folder that start with subset underscore, and then just piping that into word count -L, which is counting the lines. And so in total, we have 67 files that we're going to submit. The FASTQ format is a genomics file format, but you could absolutely apply the same principle of cutting up your data to whatever your data might be. So if you're working with a model, or if you're working with proteins, chemistry, physics, as long as you can subdivide that problem, it works very well. And so we now have this list of subsets that we've created that we can start to prepare our list of jobs from. And preparing that list of jobs is relatively simple in this example. What we want to do is we want to submit a single job for each of those subsets. And so we just have to list that set of subsets into a file. The easiest way to do that is you can do ls input /subset underscore asterisk, and the asterisk is just going to be your wildcard character. And then you're going to pipe that into xargs, just because right now it has that inputs /segment. We want to remove that for cleanliness. And then you're going to output that into a file called listofreads.txt. And what that will look like is this. We have a simple text file where you have the name of the input file for each one of your jobs. Functionally, what this is a CSV, so a comma separated value, similar to an Excel, but in this case, it's just one column. And so Condor is going to be able to use that in order to generate each one of your jobs as we scale up. And so down here, we're going to look at our actual submit script. So we now have our list of jobs. We want to do this. We want to run a job with each one of these individual input files. So we want to now look at what our executable file is going to look like. And that's that minimap2 program. And so if we open it up, and I'll open it up on the side because it looks a little nicer. If you're not doing genomics, you don't have to worry too much about it. What's functionally important here is we have these positional arguments that we're taking in through the command line, and we're assigning it to a variable. In this case, we're assigning the first position in the arguments line as the reference genome, the name of the reference genome file, and then position number 2 is going to be the name of the read file. So this is similar if you did just /run_minimap2.sh and then gave it the name of the reference file or the reference genome file and the name of your FASTQ reads. And so your script in shell, whether you're doing this in shell, Python, R, Julia, whatever program you're going to be working with, as long as you can run those position-based arguments, you can pass these variables along, and your script will go ahead and paste the value of that reference genome throughout your script, wherever you tell it to place it. So in our case, we're putting it into our minimap2 command, as well as our output. And then there's a second step, which is happening after minimap, which is the SAM tool sort. And that's just going to take the output of minimap, sort it, change file format, and output it to a second file. This is very specific to bioinformatics and genomics. But in this case, you would just be able to swap this out for whatever your workload would be. And so we now know that what we need to pass through to this shell script is 2 arguments. We need to give it a reference genome, and we need to give it a reads file. And it has to be in that order. And so Condor can actually handle that for you behind the scenes. And it does that by using what we call the submit file. The submit file is what Condor, Minimap2 is a function, or it's a program that you can install if you're doing genomics. It's not inherently part of shell, but you can install it if you're interested in doing that. So the submit file is sort of the instructions that we're going to give Condor to say, I want you to run my job with this executable, this script, these arguments, these input files, these resources, and you can describe what your job should look like. Oftentimes, this is also called something like a job template. So it's how you're describing all of your set of jobs to look like. And the most basic job submit file looks something like this, where we have a container_image equals, and that's going to point to a path, which is going to have your container. You don't necessarily have to run things in containers on the OSPool. However, it is one of the easiest ways to solve most software issues that we tend to see. One of the important things is when you're running on the OSPool, you're running on a very heterogeneous system. So you're not sure what type of machine you might land on. And if your program is expecting a very specific version of, let's say, GCC lib, and the machine that you land on is out of date or doesn't have that version, you're going to have job error issues. And so containers just standardize that environment. It's kind of like a little virtual machine that you're starting up inside the actual machine that's running your job. And so we're going to have this container image path. We're going to have our executable. That's what we actually want to run. Our arguments line, that's what we want to run as the commands for that executable. And then we're going to have our transfer files. And so in the OSPool and with Condor, you have to transfer all of your input and output files declaratively. So in here, you're going to tell it, I want to transfer these files, or I want to transfer these directories. Condor will pack those up, move them to the execution point or the compute node, and then when your output files are done, when your job is done, you can tell Condor, I want you to bring back these files. While you can transfer files without transfer output files, it'll just bring back whatever is at the top level directory. So your current working directory in your job. One of the benefits of using this transfer output files explicitly is you're always going to know which files are coming back. And so if you know that you need this specific file because it's what you're looking for as your output, and your program happens to generate 80 files per job, you don't have to bring back all 80. You can bring back one. You can also map or remap your files. And what this is Condor will initially try and bring back your files to wherever you submitted your job from. So in this case, it would be this tutorials-minimap2. But you can imagine if we submitted 70 jobs, you're suddenly going to have 70 files in this directory, which is not going to be fun to parse through. And so we have this neat output directory. We can just do a remap, and I'll show you a little bit more as we're going through the syntax for that. But remap, you can imagine it as saying, Condor, I want you to take this file, and I want you to move it to this directory or move it to this new name. So you can do both a renaming and a moving operation all in one command. Next, we'll have this logging information. That's just to capture your log info. So that's going to be your error out and your log paths. And then down here, we have our resource requirements. So that's where you're going to tell Condor, I want you to request 1 CPU, 1 gig of disk, 1 gig of memory, whatever you might need. And then all of our submit files are going to end with this statement of Q. And Q just tells Condor submit 1. So we have this template, but it's missing all of our information. So if we actually look at what our run minimap2.svb or submit file is going to look like, it looks something like this. And I can open it up also on the side, which might be a little cleaner.
Danny, can you make your screen just slightly bigger?
Yeah, like zoom in.
Yeah, so like Command plus. Yeah, perfect. Thank you.
Thank you. So we have our container image path, and you'll notice that our container image is pre-appended with this OSDF prefix. That's because we're going to be using the Open Science Data Federation to move our container throughout the network. The reason that we do this is that container is going to be used over and over again across all of our jobs. So it makes sense to cache it as we pull those jobs through the system. We have our executable. In this case, it's that run_minimap2.sh file, and that's what we were looking over here at. And we have our arguments. In this case, I've hard-coded for this specific job, and this is a one job test job. We've hard-coded the names of the files. So in this case, we're telling it for the first argument, I want you to use this reference genome.fasta.mmi file. And then for the second position, I want you to use the subset underscore AB blah, 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 fastq. And so that's going to be our subset of reads. And so functionally what this will do, or what Condor will do, is copy this and append it just to the end of run_minimap2.sh when it lands on a machine. We also have to transfer in our data. So our reference genome is also going to be something that's going to be shared across all of our jobs. So it also makes sense to place it in the OSDF directory and use that OSDF prefix so that we can cache as our jobs go through the system. However, our reads are going to be changing. So every read is only really going to be used once. So that's why we're keeping it in this other input directory rather than our OSPool directory, because it's not something that needs to be cached. And it can be transferred much more quickly throughout the system using a standard SCP command, which is sort of what's happening under the hood with this, rather than having to cache it first. And so Similarly, we want to tell Condor, hey, I want you to bring back this specific output file. This program will actually generate a handful of files, about 6 or 7 per job. And rather than cluttering up my entire directory, I just want my output file. And so I'm telling it, bring back this BAM file. And I want you to take that file and remap it or rename it and place it in the outputs directory. And so the syntax for this is somewhat simple. You just take the name of the file that you want to change or place somewhere else. And you do the name of file equals and the directory or the path that you want to put for that file. In this case, we just want to put it into outputs, and then we put the full name of the file. Down here, we're capturing our error, output, and log. You'll notice that there are these special variables called cluster and process. Each one of our jobs is going to have a unique process ID. But our set of jobs, every submission, every time we submit a job or a group of jobs is going to get a cluster ID. And so this is a really good way to keep unique names for these output and error files, which are very easy to overwrite. And so we use the cluster ID hyphen and or underscore the process ID to identify between them. However, in the log file, we only really want one log per submission. And so we can just use the cluster ID to have a unique name between submissions, but we don't need a log file per job itself in a multi-job submission. And so that might not make a lot of sense right now because we're only submitting one job, but as we start to scale up, I think it'll become a little more clear. Here, we're going to express our requirements. So we're asking for just 2 CPUs, 5 gigs of disk, about 10 gigs of memory. And we're going to submit one job. And so in order to submit your job, it's fairly straightforward. So once we've written out that command, now as you're going through later, if you are more interested in understanding what each of these options are, we have a little bit more in-depth verbiage here of how it's working. But if you come down here, you'll see this condor underscore submit option. And that's the condor command to actually submit your job. And so if we go ahead and run that cell, you see it says we're submitting one job with the cluster ID 1. And so I'm going to open up my terminal so we can have a more live view version of it. We have this really neat command called condor underscore watch underscore queue. And so this is a live update of how your job is running. It updates every second. And so that job that we just submitted, it's already running for me. It's done and it's outputted and returned back to me. If I go to my outputs, I see here that I have that output file with a horrendously long name, a very common thing in biology. But we have that output back in our directory. And so we were able to submit one job. That's exciting. But for the most part, you're probably not here because you want to submit a job. For the most part, you probably want to submit tens, hundreds, thousands, millions of jobs. And so that's where Condor really shines. Condor is able to take in that list of jobs that we created earlier. If we come back here and submit that same job for each one of these entries on this list. And so the way that we can do that is Condor uses variables similar to Bash. And so in Condor, you can set up custom variables. In our case, we're going to be using the variable reads underscore subset, but you can name this whatever makes sense for you. And so in our submit file, if we edit our arguments line, and we want to sit down here and think about what are the lines in our submit file that are going to be changing between all of these files, usually it's going to be anywhere where we're referencing that input file, that reads file that we're giving it. And so the places that we reference it is our arguments. So we want to actually put the name of that read file there. So we can use this reads underscore subset. surrounded by parentheses and a $sign. We're also going to have to transfer that reads file from our inputs directory. Our reads input file name is also appended onto the output file. This is a common trade of biology. But if you don't actually append the name of your reads file into your particular job set, you might not need this variable there. In our case, it's going to be there. We're just going to place a variable in the output file. So we have that mapped, the name of the input file, reads, all throughout, as well as our output remap set. Lastly, in our output and error log and our output and error files, I usually like to add our read subset file name, because as I'm going through, job cluster 1 process 43 might not mean anything to me, but I know what this file is. And I can go back and say, oh, this file had an issue. And so just from the name of it, I can quickly see, there was an error with process 47. It maps to this file. Did I just delete that file by accident? That's usually the case. And so it's a good way to, or a good habit to have. And so we have now this reads underscore subset variable all over our submit file, but we actually have to assign a value to it. And where we assign it is using our Q statement. And so the way that Condor works is using this Q statement, you can take reads underscore out or reads underscore subset and assign it a value from your list of reads.txt. And this functionally works very similar to a while loop, if you're familiar with those, or a for loop, where you're going through this list of values that are the list of the reads that you want to go through. And for every one, you're going to replace the value of reads underscore subset with the name of that, or the value of that line. In this case, it's the name of that subset file. And anywhere where you have reads underscore subset, we're going to essentially copy and paste subset underscore AB, so on and so forth, all throughout our submit file. And then we can submit a multiple job set. Now, before we submit 67 jobs in this smaller cluster or smaller pool, we want to make sure that we test. And this is one of the big things that we usually tell our users. Submitting a job, single job, is generally fairly easy. Most of your errors are going to happen between 2 and 3 jobs. If you can submit 3 jobs, for the most part, your workload should fly through. But beyond just finding errors that might be happening with your submit file or your SH file, it also gives you a little bit of more data about what resources you're requesting. So in our case right now, we are requesting, if we go back to our original submit file, we're requesting 2 CPUs, 5 gigs of disk, and 10 gigs of memory. This is something that I sort of just guesstimated, had an idea of what my files look like, and said, let's try here. Now, easiest way to test out a couple of your samples is just do a subset of that list of reads.txt. You could either copy and paste it over here, or you can use the head command and say, just give me the top 5 lines, and output that to test set of reads or list of reads.txt. And so if we look at that file, it just has these 5 lines. It's the first 5 reads that we're going to test. And if we look at our submit file, our new submit file, it looks something like this, where we have most of the things here have stayed the same, but we now have that read underscore subset variable throughout the file. We're also just giving it that shortened list of input reads, or that shortened list of jobs that we want to give it, just to test out our soon to be scaled up job set. And so we can submit our job, again, using Condor submit. In this case, just for simplicity and making it easier, we've made a second file, and that's run underscore multi underscore minimap2.sub. And we see that we've submitted 2 jobs, and now it has a cluster ID of 2. So if I go back here into my terminal and say, show me my jobs using Condor watch queue, we'll see that right now 1 job is inputting, it's transferring in, it started to run. 4 of them are currently trying to match to a machine. And as those jobs process through, one is finishing up, the next one's rolling in. You're seeing that we're getting about the throughput of maybe 1 job at a time, which we would want to have higher throughput. And I suspect right here, the problem is we're probably just requesting too many resources. In this sort of setup of the pool, and this is not the full OSPool, obviously, we're just asking for too much. So we have fewer machines that we can match to. And so as those jobs start to come in, we'll go into our outputs and see, yeah, files are getting generated. And so we can also check on our logs. And your logs is going to be a really useful set of information. Right now, this looks all like gibberish, but what you're usually going to want to look at is this table down here, and it tells you what are your resource usage. In our case, this first job, we requested 2 CPUs. We use 0.8 of a CPU. So we're requesting way more than we need. We're requesting a lot more disk than we're actually using. Same thing with memory. And so this might be a good place to start tuning our resource requirements. We can look at another job here. We're using about 1.25. So I might be a little bit more inclined to leave the 2 CPUs, but again, we're still over allocating memory, we're over allocating disk. Same thing for the next job. So there's a little bit of variability, and that's expected with your data. But we're seeing that memory and disk are just getting underutilized. And so we check on our jobs. All of them are done. We've gotten all of our output files. And so now we're going to actually scale up to the full production. And what we've seen is memory and disk are just using way too much. So for our full production, we might want to cut that down a little bit. And so, again, you're always going to want to read your log files. It's going to have useful information on how efficient you're being with your resources. And the more efficient you are, the more jobs you can start up at once. So now we're going to go to our full job set. In our case, originally we were asking for 2 CPUs, 5 gigs of memory, or 5 gigs of disk and 10 gigs of memory. Based on our results, we're just using very little of it. So a good place to start is let's lower this down. And for most jobs, we're going to encourage you to start small. We want to have you start as small as possible. If your jobs go on hold, we can go back and edit those requirements and re-queue them when they run out of memory. But you're going to get a lot more throughput starting small. And so we want to change our submit file, our run underscore full underscore minimap2 sub file, so that instead of using 2 CPUs, 5 gigs of memory, or 5 gigs of disk, 10 gigs of memory, we're just using 2 for all 3 of them. And so we're going to open that up right here. This is that submit file. It's the same thing as our previous test, except now we're using the full list of reads that we want to map, not just the 5 subset. We want to change this to 2 gigs disk and 2 gigs of memory. And so I'm going to save this file. And so now if I run this cell again, we should see these values change back to 2. And so this looks good. So now we hit Condor underscore submit and submit this new file, and that's going to run through our full data set, all 67 jobs, but using fewer requirements, we're likely to see we're running 67 jobs, cluster ID number 3. And so if we check our queue, you now see that we're starting to input 4 jobs at a time, where before we were only processing 1. And so that's one of the key aspects of using the OSPool. The smaller your resource requirements are, and that's why we spent a little bit of time breaking down our data, the more machines that we're going to be able to match you to, and the faster your jobs are going to process through. And so you'll see these jobs continue to process through. They'll kind of run at about 4 to 5 jobs at a time. And as they come through, right now they're all transferring. Some of them are starting to run, and they'll start to finish their jobs, and you'll continue to get your results. And so at this point, you've started to scale up your research. You've started to submit now full data sets. This is using a small set of the humpback whale genome. It's only using one of its 32 massive chromosome sets. But you can imagine running across the full data set that that student would have of millions upon millions of reads. Using the OSPool would let them scale this up very easily in about an hour. And so for the next steps, and what we like to remind folks is we're always here, facilitators are here, and we always want to chat with you about scaling up your research. This is what really gets us excited. So if you're kind of hitting that wall of you're like, I need to scale up my work, come chat with us. We know some of these tricks that we can help you subdivide your workflows and run them in parallel so that you're getting a lot more throughput. All right, with that, you can continue to let the jobs continue to run through, or you can also kill it if you want. But I think Christina's up next with a little bit more information about using the Open Science Data Federation.
Yeah. Hi, everyone. I dropped in a little late, but I'm one of the facilitators for the OSPool, along with Amber and Danny. So I'll just wrap up Danny's section by saying the infrastructure you're using today, and may have already been said, is these guest notebooks are not accessing the full capacity of the OSPool. So a next step, if you're looking at this and are like, this is for me, would be to apply for a full account. You can still use the same kind of Jupyter interface, but behind the scenes, you have access to the thousands and thousands of computers that are a part of the OSPool. So we'll.
Oh no, bad internet has.
I know. I was like, Danny and Amber stopped moving. Ah! Okay. We'll hope for the best moving forward. Okay. So you all just heard a lot about scaling up your computing, and hopefully this is a very specific example, the mini map read mapping exercise. But if you have any kind of problem where you have lots of chunks of data that you need to analyze, hopefully you can see how that would map to your to what you're doing, and what it could do for your research. So we're going to shift gears a little bit now to talk about data a little more than computing. Obviously, the two go together, but we want to focus on how we support data distribution, both alongside the OSPool and computing, but also just in general as part of our OSG services. So I'm going to start with a slide about how people might identify in the research data ecosystem. So some of you might be data providers. So you're generating data or curating data, and you want a certain audience to use it. You want to share it. And that audience could be the public because you're like publishing something, or it could be your collaborators, or even just you. So you want to have it available. And then some of you may be data consumers or users. So you want to find data and download it and use it as part of your analyses. And a lot of you might be both of these people at the same time, right? Where you're getting data from somewhere else, analyzing it, and then sharing it, or you generate data and you also analyze it, right? So the point is not so much like which of these is you, but to recognize that these are two kinds of roles or identities that exist, and we want to help support both aspects of this of working with data in this way to support your research. Okay, so how do we do that? We have a service called the Open Science Data Federation, and it's a network that connects different data repositories into this national data ecosystem. And so it's both a way for data providers to make their data available either publicly or within smaller groups in a lot of different ways, which we'll talk about in a minute, and for people to get data and use it for their work. So we have this network, this federation. The software that's used to run it is called Pelican. So it's the little Pelican logo right there at the bottom. We like our birds in the OSG and Path projects. So that's when we talk about the OSDF, this is what it is. I'm going to talk a little more about it in detail because it's kind of general and hopefully help people understand. So first, we're going to put on our data provider hats and be like, I have a data set I want to share via the OSDF, for example. So the data is already stored somewhere. Maybe it's in S3, maybe it's in a local storage service, maybe it's stored somewhere and available through Globus. So you have as the data provider, you have the data somewhere. And so what we're going to do is add this Pelican functionality, this Pelican thing called, it's a service called the Origin, which has a namespace, which is like a label for the data space attached. And this is something that can just plug in to existing data stores. So in each of these different options, we can set up a Pelican Origin service with an associated namespace. And then those origins are integrated into the overall network of the Data Federation, the Open Science Data Federation, that also connects them to a few central services that help like coordinate where data should be and where it goes, and some cache services. This is kind of what Danny was describing earlier, that make data download more efficient if you're trying to use it at scale. So the message of this slide is if you have data you want to share, you can plug in this Pelican Origin and a namespace, and then it integrates into this network. Okay, so then how do people access it? Well, there's a special URL format that people can use. There's a couple tools that we'll look at that allow you to download the data. So the overall format is Pelican instead of HTTP, the address of the data federation, the name of the namespace, and then the actual path to the data you want to download. So in this example, if we had data in our local file store, here's the origin. Our namespace is rad bacteria. And one way to do the link would be Pelican, the OSDF. If this was part of the OSDF, data federation is this URL. Then our namespace is rad bacteria. And then we have the path to our sequencing data. For the OSDF specifically, there's a shortcut URL where you can combine these first two pieces into this single OSDF://. But all that the person using the data needs to know is this information, is what's the namespace, what's the data federation, what's the namespace, and what's the name of the file. And then they can access that data in a couple different ways. So they can use a command line tool, the Pelican command line tool, to upload and download and look at things. There's a Python library where you can just load the data directly from the URL. And then, as you've already seen a little bit, if you're using htcondor to submit jobs, you can pull the input data for your jobs directly from an OSDF origin into your job, and you can actually put output data there as well. So those are kind of the different options. Part of the reason we're talking about the OSDF is I believe some of the storage at least the University of Hawaii has an origin and a namespace associated with it. So it could be possible for some of you to access your data directly using some of these methods and tools. So that's something to keep in the back of your mind as we proceed. Okay. I'm gonna do another demo. For those of you that have your notebooks up still, you can follow along or you can just watch. If you have questions, drop them in the chat. Amber and Danny, are there any questions we should
Nothing so
address?
far.
Nothing so far. Okay, great. So again, for folks, if you don't have the notebook open and you want to, here's the link. You should be able to log in. But if you have it open and you have the read mapping tutorial, then we're going to go to, if you're not on the main file browser on the left, you can go to that and then go to the tutorial OSDF NOAA section. And then there's a couple notebooks that show that'll demonstrate ways that we can interact from the OSDF using these different options, the command line, Python, and so we actually don't need to run the setup step, but I'm going to open this just to explain a little bit about the data set. So for this example, we're going to be using a data set that is in S3 storage and has had an origin and a namespace put in front of it. It's a data set from NOAA. It's basically a bunch of weather stations and their historical weather and climate data. And so the namespace is this long thing. So this is a longer namespace. Usually they're not this long. And so it's integrated into the OSDF. And let's say the first file we want to download is the list of stations. So the full URL is going to be OSDF : / namespace and then file name. So that's what we're gonna use this example and a similar one in the 3 notebooks that follow. Okay, yeah, that's it. That's it about the data. But again, this is just an example. These principles should work for any data set that's in a storage somewhere where there's a Pelican origin and namespace that are connecting it to the Open Science Data Federation. Okay, so we'll just go through these real quick. Boom, boom, boom. Option 1, you can use the terminal command line to download data and look at things. So almost all the commands have the same format. Pelican, object, whoop, can't double click. Pelican object, and then some kind of function, and then usually the URL that you want to use. So if I want to download the station list, I'm going to do Pelican object get is the action, and then the URL that we just talked about, the special OSDF Pelican URL, and then just a note that I want to download it to my current folder. So if I run this cell, either Shift Enter or play button, it should appear on the left. Here it is. And we can see that this is a list of all the stations, their ID numbers, latitude, longitude, codes. These are like airport codes. And we've gotten the data. So this next section just shows basically the same thing, but a different file. So if we wanted to download a specific weather station data, we'll use the same beginning part of the URL, but the path to the data is different. It's in these subfolders. And then we use the station ID as part of the file name. But we use the same command, Pelican object get, new URL. Da-da-da. This should also appear on the left. There we go. And we can see this is the weather data for this particular weather station, which happens to be where Amber and Danny and I are in Madison. So that's option number one for getting data from the OSDF. So if all you need to do is upload, download data, you can do that with this command line client. And this notebook goes on to talk about sharing data, listing available data objects, and installing Pelican to your own computer, because you could run it anywhere, Mac, Windows, Linux. Option number one. Option number two, this is a Python thing, so that's what I want, is you don't even need to download data from the OSDF to work with it. So we're going to use the same weather station URL. And here we have some code that runs Python based. It's going to analyze our weather station data and produce a plot. But what we could do instead is we could remove the local file. So it should disappear. Disappear. Maybe. Okay. In a minute. And there it goes. And instead, we could construct this URL. So here's our OSDF. Here's the station ID. And we can just read that in using a read CSV function. In this case, it's Python. Nope, you got to define it first. Got to run my cells in order. Okay, there we go. It's loaded in. There it is. And then I can do the same analysis as before. So this might be good if you're working with data that's maybe too big to download locally, or you just don't want to. You want to be able to pull it directly. It is at the moment, this kind of streaming functionality is only available in Python. So limited time and effort. We don't have implementations for every language, but it should work in a lot of Python different libraries. Here we're using pandas, but it should work for others as well. Okay, and then the last example, this I'm not going to do in a lot of detail. These tutorials are preloaded in the guest notebook, so all of you can go back to them anytime and try running them and going through them in more detail and reading them. But basically, this is the same idea as the read mapping tutorial, just a different use case. Instead of a bunch of FASTQ files with different read subsets, we have a bunch of weather stations. So if we wanted to run our Python script on each station, it's like 120,000 plus. It's maybe not something we want to run on our own computer, but we could submit this as individual jobs, one job per weather station, and send them out to a resource like the Open Science Pool. Now, in this particular example, we're not going to go crazy and run on 120,000. We're going to work with a little subset, but the principle is the same. You know, you could use the full station list instead of this abbreviated one. So this cell will basically cut it down so that here's the list, you know, a little more manageable, 10 stations. So we're going to submit 10 jobs. And then this job template, very similar to what Danny was doing earlier. We're going to specify software environment, our inputs, information about the jobs, resource needs. So this is going to look hopefully a little familiar to you all. Container. And here's where we're going to use the OSDF. It's just in this, well, two places. One, we're pulling our container, but also we are constructing the URL for our station and putting it here in transfer input files. And we're using that same trick at the end of station ID is a variable that'll be different for each job, and it's being created or populated from our station list file that we just looked at. So 10 items in the list, we should have 10 station IDs, 10 jobs. So if all goes well, that is indeed what happened, which is great. And we can use the same tools, Condor Q, Condor Watch Q, to look at our results. Let's do Condor Watch Q because it has fun colors. Yeah. So yeah, here we're transferring our input data. Hopefully these switch to running soon. And again, this is just 10, but from our perspective, there's no difference between like 10 and 10,000 in terms of how easy we want it to be to submit that many jobs. So there we go. Also, this analysis is really fast. You wouldn't need to necessarily split it up one thing per job. But if your analysis took more like 15 minutes to an hour even per thing, it would be worth it to split up. Okay, so I saw some questions in the chat, which I'll answer. Are there any questions in a minute? Are there any questions about that was kind of a whirlwind tour of how to use the OSDF? But I think the takeaway, can folks see my slides? Yes, okay. The takeaway is kind of like, you know, could this work for you? Do you have data that you either want to share in a specific way as a data provider? Is there data you want to access that might be publicly available, but you don't quite know how to get to it? Is there data you want to process? This data federation is meant to help people solve those problems. And as this says, there's OSDF data origins at Hawaii. Maybe, you know, if this is something you want to do, you can work with the staff there and us to make this work for you. And for some of you, may be like, this doesn't, I don't have a lot of data, or I can just email things, or whatever. Then that's fine. We want to help people who have a data problem where this could be a good solution for them. So to answer the question that was in the chat about OSDF, so there's two answers. If you apply for the OSPool and you get an account on one of our access points, you automatically get your own personal space that you can use in the OSPool alongside the OSDF. So like Danny and Amber and I all have accounts on OSPool access points, and so I have a Christina space that I can use to store data, and it's just accessible to me, and we can support projects as well. Separately, there's people outside, like people at Hawaii, or like University like Hawaii, or who else is sharing data? I think like Purdue has some storage and Missouri has some storage. There's different universities that have storage that's now connected to the OSDF. And some of it has data from their own researchers that they're sharing. And some of it, they have told us at OSG that we can find other people's data to put there. So that process, if you're like, I have a huge data set, I want to share it kind of outside my normal OSPool account, there's a different application process for that. And if you came from Hawaii, we might be like, well, let's start with Hawaii first and see if there's space, if the right space to put your data is there. We don't actually own any of the storage. So remember, there's a distinction between the storage where the data is and the Pelican origin pieces. So we are relying on different people to provide storage, to share their data, and then we're helping run the like OSDF sharing network layer. So the Open Science Data Federation in some sense doesn't provide like actual storage. We just help people who have storage and data share it in different ways. But we can also connect you with like members of our network who do have storage and make that happen. Yeah, so those are great questions. Okay, so that flows nicely into next steps. So things you can do. You can continue to use the guest notebooks that Danny and I were demoing today. Anytime, try things out. If you want a full account with access to the full OSPool, I think the form was dropped in the chat. Fill that out. We'll schedule a meeting. We'll get you set up, and you'll get access to a full access point. more persistent home directory, the full OSPool, and space behind an OSDF data origin. So lots of features. If you think this would be useful to you, I think it's worth checking out. We are here in general, the facilitation team, to help figure out if things are a good fit and what you should do next and help you solve problems and get started. So you can contact us via email. We have Zoom office hours. Our Tuesday hours are at a reasonable hour for all of you. Thursday would be a little early. We do trainings also around this time of the day on the third Tuesday of the month. So we actually have one next week, and we have a bunch of user guides online. So those are all resources for you to connect with us and to learn and to implement your workflows. If you're really like, I love these people, I can't get enough of them, we have different community events typically in the summer that folks are welcome to join. We have a general conference called HTC this year 26, which is going to be in early June in Madison, also has a virtual component. This link has the save the date. And then probably more applicable to all of you, we have a week-long summer school, typically in right now, we're thinking early July. We bring people to Madison, we pay for travel, and we do a week-long school intensive where we try to get you up and running on the OSPool within the week. And applications typically open in February. We try to publish widely, so I'm hoping most of you will hear about it in some capacity if you're at all connected to us. And it's a really good time. So I think if you are really interested in learning more, or you like get started and you're like, I really want to take it to the next level, that's a really good event to consider. So I think that's actually, yeah, that's all the slides. Thanks to the NSF for funding a lot of our services and activities. And I'll actually stop sharing and see what questions people have. Yeah, Gavan?
Yeah, great stuff. Thanks a lot, you guys. That was really cool. No, I had a question because I was using Access to request allocations from OS, and I was looking at it. I had all the allocations from Jetstream, but the ones from OS, it was still pending. Um, and I was wondering, like, maybe I wonder if I should cancel that and just have you guys help me with that instead.
Yeah, I mean, I think it would be good for us to figure out why it's still pending, because you can technically, you should be able to get an allocation in theory through Access, but
Right.
you shouldn't let that hold you up. You should feel free, anybody should feel free to just contact us directly through that
Okay.
application form we shared, and we
Okay.
can just proceed with that, even if we're still trying to figure out why
Yeah.
that is still pending. And if you want to reverse it, that's
Yeah. Yeah.
fine.
Okay.
That won't impact your access directly through us.
Okay. I'll come bother you guys later then. Thank you.
Yeah. Okay. I'm browsing the chat for any other questions. Folks, feel free to raise hands or just shout out if you have things. So we talked about applying for the OSDF, storage. Yeah, if you have analysis, the account request form is definitely the place to start, because then we'll meet and talk about your work and how to get going. Yeah, any other questions?
I guess just with reference to the first tutorial, so if we don't like provide one of those variables in an output file name, then it's just going to append everything into a single large file. Is that right? No, it's going to
Overwrite.
overwrite. Okay, okay.
Yeah, no, that's a very, very important question and distinction.
Definitely have made that mistake before.
Okay,
Yeah.
that's good to know. And so I guess the most as you showed us, the most straightforward and traceable way to um, make different names is to include the cluster ID and the process ID.
Yep.
Yeah,
But
that's
no,
always going to be a unique combination. So it's pretty foolproof in that way. Yeah,
thank
I
you.
will say the mindset, like the fact that we overwrite and don't append, does mean that our approach or implementation of high throughput computing generates a ton of files, or it can. And so that's why I think the example Danny did has a lot of good best practices in terms of naming and organization, so that you set yourself up for success.
I also think it's a good time when you're starting to get to that point of, oh, I'm about to submit a few million jobs. I can tell you my own experience. I submitted 50 million jobs throughout my PhD. It's a good time to have that check-in with a facilitator because we can go through that submit script and make sure that, like, all right, you're going to submit 50 million jobs. That means you're also going to generate 150 million files. Let's think about where we put those files or what we need to do in the background to make sure that those jobs don't hurt any of the systems, that your jobs process through perfectly, don't have infrastructure issues that come about. And so that's a great time to either drop in on office hours, shoot us a message, schedule a call with us, just to have a check-in.
Okay, thank you. Yeah, that's good to know. Well, if that's it, then I'm going to go ahead and stop the recording and thank you all for um, running this training.
Yeah, thanks for having us.
Yeah, thank you, you guys.
Yeah,
Great
yeah.
job.
Hope it was useful, and we're always happy to chat.